Hi, this is Miss Litton, and this is my wonderful Period 1 AP Biology class. Say hi.
Hi.
All right. They don't sound excited. And this is Chapter 13, Regulation of Gene Expression. Just look at that title. That's huge, okay? Because our code book is what? DNA. And what we're going to be talking about today, obviously there's so much more than what we're doing here, right? But what we're talking about today is how you are you. What genes are turned on and where they're turned on and how long they're turned on. And we'll start talking about bacteria because that's easier to understand, and then we will level up. And then when we get into chapter 14, we'll talk about some other kind of epigenetic forms of control as well. So the next two chapters are pretty much linked together. We started talking about this last time, so I'm going to move quickly through this area. A little review. Could my youngest bio buddy very quickly just go through this review, talk about what this is about? Go ahead. It should be easy for you. Not details, big ticket items.
I was right. You know what said incomplete dominance? I wrote about the multiple allelic traits, but then at the very end I put comma.
All right. So we understand to get the message out, we form mRNA, and then you use that mRNA to build a what? Protein. mRNA to build a protein, yes? Okay. So the question is, if all of our body cells have the exact same DNA, why do we have different proteins, right? Why is different parts of that DNA expressed differently in different cells? And that all has to do with which genes are turned on and which ones are turned off. This is called differential gene expression. Every cell has the same DNA, but what aspects are turned on and available for translation, transcription and translation will be different in each cell. So we talked about this, right? Or this was period 3 that I talked about? We talked about it, okay? So we talked about that there are all different areas where you can exhibit control over the process. Ultimately, you're talking about this, a fully functional protein. And we know that the DNA, which is here, right, that has to be transcribed into mRNA, and then you use that mRNA to build a protein. But all along here, there were levels of control. And where were some of those levels? Tell me. Transcription. And that right there is the biggest level of control. And we're going to spend a lot of time today talking about that. Okay, where else do you have control? The processing. What is processing about? Putting on a cap, tail. What else? Cutting out introns. That's huge. Because depending on which pieces of the DNA you remove, you can affect what sequence of genes you have, right? Okay. And then where else? Translation. How long? Because remember in translation, the mRNA is hooking up with the small and large ribosomal subunit. So can it get translated? Can it hook up with the ribosome? Is there some small interfering RNA that's binding to it so the RNA is not single-stranded anymore, it's double-stranded? Because if it's double-stranded, who can't land on there? tRNA. Okay? So there are all aspects of control there. Where else? Longevity, obviously. How long till it's digested? Where else?
When it comes out.
Okay, when it leaves the nucleus, whether or not it's able to go through a nuclear pore, and where else? Come on. At the end? I think you're the one who said that last time. Remember we talked about the folding pattern, right? The primary structure was dictated by the DNA. Remember the primary structure is the sequence of amino acids, right? Secondary, what are our options on secondary structure? Beta pleated sheet or alpha helix. What's the tertiary structure? Folding pattern. And that's due to the what? R groups. How many different R groups do we have? 20. So that folding pattern, that's done by what kind of proteins? Chaperone proteins, yeah. That work to fold it in its correct shape. And then what's the next level of structure? Quaternary. And that is when you have what? Multiple chains hooked together. So aspects of control, this is huge. looking and studying proteins and how they're made functional, very big study field. It's called proteomics, okay? So that's huge, okay? Where else do you have control? One more place in particular. Right here, you guys. Remember, DNA, what does it do? What is that called when it's all supercoiled? If that DNA is not here, right, you can't even boom it, right? That DNA, you've got to undo the supercoiling. And what if there's something on there that prevents you from undoing the supercoiling? If it's prevented from ever getting, then you can never express those genes. It's like permanently disabled, okay? And then you've got to be able to get helicase, to unwind, unzip, to even make it available. So that right there is another area of control. So we're going to start off by talking about bacteria, because that's a little bit, that's easier, okay? So bacteria live in our gut. They are exposed to whatever we eat, so they will make the appropriate enzymes that they need in order to metabolize the nutrients we provide them. So it's going to vary depending on the diet that they are exposed to. They do not want to make enzymes they don't need. Bacteria have preferred food. Who did we feed into the process of cellular respiration? What did we start out with? Glucose, right? Right? Glucose, how many carbons? 6. What are you going to do? 3. How many steps? 10. How many enzymes? 10. Right? So we started out with glucose. Bacteria would prefer to use glucose. But if there is no glucose, then they'll use other items like lactose. Where would lactose come from? Milk. What is lactose made out of? Come now. It's a disaccharide. Glucose plus galactose. Maria, bringing it home. Glucose plus galactose. Yeah, makes lactose. What's the enzyme that would digest that? Lactase. So you don't need any lactase unless you have what? Lactose and no what else? Glucose. You only want to make that enzyme if your preferential food is not available and you do have lactose. Otherwise, you want those genes turned off. It is a waste. Yes?
So say I never drink milk and then one day I drink milk. How long will it take these things to make the lactase? That's totally way off.
Yeah, no, I mean, it would probably take minutes. I don't know. But that's okay. I did the poll.
What prompted you to drink the milk if you never drank it?
Let's say I'm 70. Okay, now, moving on. Okay. I love you. All right, now, how many different amino acids are there? 20. Now, tell me about amino acids. Some are referred to as being what amino acids? You know what they are? Starts with an E.
Enzyme.
Oh, you're so essential. Have you heard of essential? Yeah. Some amino acids we can synthesize. We have the ability to build that amino acid if we need it. Some we don't have the ability to build. We don't have the enzymes to make the amino acid. We don't have that for some. Those are your essential ones. Those are the ones you need to get from your eating, from your diet, from your food. Okay? So bacteria have those abilities too, to make certain amino acids. They have the ability to make certain enzymes. All of that is going to be stored in their what? DNA. So they can turn different genes on at different times. So when you look at this strand of DNA, they just put a color film over it so we could kind of separate out the different aspects of it. Now, if you look right here where the hashtag is, kind of, that just means that this is farther upstream. It may not be right next to these genes here on the right. And this is a regulator gene. What do you think it makes? I know, a regulator, okay? So this gene, if transcribed, what does it mean to transcribe something? Yeah, you're going to make what? Yeah, mRNA. And if this mRNA is translated, what does that mean? Protein. This is the protein. When this regulator gene is transcribed into mRNA and translated, it makes a protein. In this case, this protein is a what? Repressor. And it is active. It is good to go. As soon as it is made, it is functional, it is working. And what it tends to do, or what it does, is it binds, and the region is called an operator, where it attaches on. And that's that purple region here. And the reason why it was named an operator, and this is referring to the lac operon, we're going to talk about that as well, is this operator controls what happens further down, these structural genes. Because we know already that when you do transcription, RNA polymerase, what does it do? Binds to the promoter, okay? So the RNA polymerase wants to land right here, but RNA polymerase cannot land here if there's a repressor on the operator, because that repressor actually blocks some of the what? What is that repressor blocking? Some of the promoter. Remember we talked about sometimes you're on a, flying on an airplane and somebody doesn't quite fit in their seat, okay? Oh. And so they should buy 2 seats, right? But they don't, so that kind of interferes with your ability to sit in your seat, right? Okay, so we have some spillage here, okay? And so RNA polymerase can't get on this promoter, so this operator, by just controlling the operator, you're controlling all of these several genes downstream. And the reason why they named it that is it used to be, when phones were first, getting developed, you couldn't call directly to somebody, you called to a town. One line came into that town. One line came into that town, and you would tell the operator, I wanted to speak with the Jones family. The operator would then take her cord, plug it into the line that went to who? The Jones family. So she was the control point for any communication in that whole town. So that's why they named it the operator, because people then knew what that referenced and the importance of the operator. Okay? So regulate this portion right here, okay? This is referred to as the operon altogether. This region of control where RNA polymerase binds and the structural genes downstream are referred to as the operon. So when we look here, we can see the DNA, we can see the regulator gene. What did we say the regulator gene did? What does it do? It codes for a repressor, okay? In this case, this repressor right here is active from the get-go. If this repressor, where does he like to bind? The operator. And so the operator, this is where the repressor binds. Now, RNA polymerase, we know, what does it do? Binds to the promoter. Now, if RNA polymerase wants to bind here, he can't. Okay? He can't bind here because who's getting in his way? Repressor. It's not on him, but there's some spillage, right? Okay, so he can't get on here. That's okay. We must not need these genes down here. If we don't need them, let's not build them. Okay? But then you start eating some lactose, and you have not eaten any glucose, so all of a sudden there's all these lactose right here. Okay? So when the lactose is here, the lactose, there is a binding spot on the repressor for the lactose. The repressor can only hang and hold hands with one thing at a time. So now if it's binding to lactose, what can't it bind to anymore? Operator. So this pulls the repressor away from the operator. So now RNA polymerase can get on the promoter. And it can boom down through here, and if there's a G on the DNA, there'll be a C. If there's a C, there'll be a G. If there's an A, there'll be a U. If there's a T, there'll be a A. And this RNA polymerase will start to synthesize some mRNA. That mRNA can now connect with a ribosome. You translate that mRNA in order to build a protein. That particular protein will digest lactose. So lactose gets digested, 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 including the one binding to the repressor. So when all the lactose, when all the lactose is gone, then the repressor binds back on there. Is that okay? Yeah, because we don't need it anymore. So don't build it anymore. Yes?
This might sound like a stupid question, but why does a regular gene make the repressor in the first place if it, like...
Okay, if it wasn't there, okay, then RNA polymerase would be constantly synthesizing that enzyme
So it's kind
for
of
no purpose.
Okay, so it's kind of like the stop codon, which like kind of stops.
Yeah,
Okay.
think of it like right now, think about your bedroom. Should the light be on in your bedroom at home?
No.
Why not? You're not there. It's a wasteful. Okay? Your family doesn't want to spend that money on electricity. Cells don't metabolically want to spend that money synthesizing enzymes they don't need. So you go in there and then you have the light on. Okay. All right. Now, take a look here. I've explained this once. I would like the youngest bio buddy, you explain this picture. Go ahead.
Unless you're doing TPR. And then you're doing
Okay, now, is this operon currently on or off? It's off. Why would we say this operon is off? Because you're not transcribing their structural genes. Yes, Julia.
When we're talking about the mRNA translating to the protein that digests the lactose, is the protein like the enzyme lactase?
Yes. There's more to that name right there because there's actually several different enzymes that are built together. I think there's 3, but yes, ultimately yes. Okay, this is what's referred to as, make sure eyes are up here. I see some of you, I think you're just on your phones or doing something else. I'm not sure. This is what's referred to as an inducible operon. An inducible operon. If you say something is edible, have you eaten it yet? No. But could you? Yes. Okay. So this is what's referred to as an inducible operon. It's not on yet, but we could induce it to be on. Who's going to be the inducer? Who's going to be, come on, little operon, let's get you turned on. Who's going to do that? Lactase. Not lactase, lactose. Lactose is the inducer to turn this operon on. Oldest bio buddy, support that statement. Lactose is the inducer to turn the operon on. Go.
Wait, Miss. So
Wait, I want to ask this out loud. Okay, so, come back to me. Lactose is the inducer because if lactose binds right here, then what will happen? The repressor will come off. Yeah, you'll move the repressor, so then you'll be able to transcribe these genes. Clarifying questions? Okay, I'm going to show you some more. Let's take a look at this picture right here. Okay, I would like, whose turn is it, young or old? Who explained the last picture though? Old, young, when you explain this picture. You can do it. Go. Remember, to teach it is to know it.
I thought it digests.
Okay? Now, we can see the promoter up here at the top. Can RNA polymerase bind to the promoter? No. Because who's on the operator right here? Repressor. Now what's happened? Yeah, the lactose started to bind to the repressor, removed it, and lactose is identified as the what? Inducer. Now RNA polymerase can bind to the promoter, and you can transcribe gene 1, 2, and 3. So this operator controlled those 3 genes. So it's like if you built a wall.
Don't look at me.
Just do the math on how to build a wall. I'm going to say that again. Just do the math. Do science, okay? Oh wait, we don't believe in science or global warming, right? Or treaties. Or women being equal, apparently, because you could just. All my brown children, you should leave now, right? Okay? My Indian children, I'm sorry. Run and hide. It's a whole new reality. I think I threw up in my mouth a little bit. Okay. Did you see me Instagramming at 3 a.m.? Okay. Anyway, if you built a wall, you don't have to monitor the whole wall, even though somebody could dig under it and, I don't know, throw a rope over the top of it. If you built a wall, if it was a really good wall, like logical wall, best wall ever. I'm making my hand smaller. If it was a wall, you should only have to monitor that one section of the wall, the gate of the wall, right? That's it. So here, this operator has extreme control because it's monitoring these other 3 genes. So you don't have to worry because nothing's going to happen here unless you have a functional operator. This is used in genetic engineering all the time. I can splice in a gene, but unless I give it a control region, I can never turn those genes on. Okay? So on this part, now we get these proteins. These proteins are going to become the thing that digests, the enzyme that digests the lactose. When all the lactose is gone, then we'll switch this back off. Now let me show you a different scenario. See if you can figure out the scenario, what's going to happen before I even tell you. Use your large mammalian brain. Go.
Okay, this is
different.
So so you could, I think, like, I guess what it is, there's like an I watched it yesterday.
Now, is this operon on?
Yes.
So it's currently on. This one is classified as being repressible. Is it currently being repressed? Could we repress it? I wonder how we're going to repress it. I don't know. It's a mystery. So our repressor is currently inactive. So it's just like, I'm just watching. Okay? RNA polymerase is on the promoter. So it can move right along, transcribing, making all this mRNA, which is translated into these enzymes. These enzymes are going to synthesize an amino acid. This looks like the little goldfish, okay? But it will synthesize tryptophan. Tryptophan, what's coming up? Turkey Day. What do you have a lot of in your turkey? Tryptophan, okay? All right, now, what happens? I have made tryptophan. Now I have enough tryptophan. What would be a really logical thing to have happen next? Who does it? Who turns it off?
The repressor.
How could we turn off the repressor? Use the... What's our product? Tryptophan. Use the tryptophan. The tryptophan will bind to the repressor and now activate it. What? Because I have enough tryptophan, I don't need any more. So the tryptophan binds to the repressor and goes, let's go. And it makes the repressive active. In fact, it acts as a co-repressor. the product acts as a co-repressor, activating the repressor. Now the repressor's on the operator. RNA polymerase cannot get on the promoter, and we've turned the operon off. Turn the operon off. Okay? So let's what? Yes?
Is tryptophan the only one that does this?
No. Now, this, anytime you're dealing with a repressor, that's underneath the guise of negative control. Okay, so I just want to help you, for those of you who've watched some and you've prepared and looked at this. Negative control always has to do with a repressor. There is something called positive control. Positive control is not about repressors. It's about facilitating and helping the process. There are things called transcription factors that help RNA polymerase bind to the promoter. So for us as eukaryotes, we're more about the positive control. We don't worry about having repressors everywhere. Our genes won't get transcribed and translated unless we have somebody to come along and help RNA polymerase bind to the promoter. No help, no transcription. Okay, that's positive control. I'm all about the help. I'm the Boy Scout, helping the little old lady cross the street. Okay, yes, before she lost her insurance. Go ahead.
Inactive repressor, how do you, and if once you have the like tryptophan or whatever, like into it to make it active, how do you turn the operon back on?
Okay, so it's turned off right now because there is what? Tryptophan. If the tryptophan runs out, if it gets consumed in some anabolic reactions where you're building proteins, right? If all the tryptophan is gone, no more tryptophan, there's no way to make the repressor active anymore. So again, he's like, oh, okay. And he stands aside again until a whole bunch of tryptophan is made, and then he gets activated and he shuts it down. Okay? So it's going forward until you shut it off. So some operons are always, usually they're left in the on state, okay? And some operons are left in the off state. You can think of like, if you want to think about electricity analogy again, your refrigerators. You leave those in the on state, right? Because you always want your refrigerators to keep everything cold, okay? It's not like just before you use it, you get it cold. It's always on, right? Okay, yes?
Okay, so just to clarify, the repressor isn't active until the tryptophan binds to it?
Yes, and the tryptophan is then classified as a co-repressor, okay, as a co-repressor. So now let's compare that. Let's compare, oh, whoops. Okay, whose turn is it, old or young? Old, explain this right here, what I wrote down here below to help you.
Start the reaction. That allows it to operate. Yeah, that's the start the that was because once for the other one, if you put in the inducer, then the protein goes away if it stops. But this one, I think if you add
what happens when your refrigerator is cold enough? Do you want it to keep getting more cold? No. What happens? It stops, right? It stops chilling. That pump turns off. And then as you open up and you lose some of the coldness, right, then it will turn back on, right? So you can think it's usually in the on state. That's a light you want to keep running. Yeah.
So would this be in every single situation where like mRNA is being translated? And
Yes.
So tryptophan is in like every single situation?
Well, no. This is just to code for the synthesis of tryptophan.
Okay.
So
okay.
absorb this.
Got it.
Right?
Got it.
Every reaction needs an enzyme. So if you control what enzymes are transcribed and translated, right, which is what's happening here, then you control what product you have. So every enzyme, there's something that's controlling whether it's generated, and if you have that enzyme, that controls whether some other product is made. You see, when you are a gigantic bag of chemicals, if you do drugs, you could I know you sell crack at nutrition. I see you. Do you see if you're a giant bag of chemicals, when you do something where you add in a chemical that's you have not evolved to handle, that could be a problem, right? And you could throw everything off. Okay. So when you think about this, it's amazing to me that we can manage so well what? Homeostasis. I mean, when you think about all the ways our body self-regulates, right? Just think about every enzyme has to be synthesized and made at the right time. Stop eating in class. Okay, did I see a question? Yes.
So if your repressor is inactive, and then just like you're not making that tryptophan, if it's synthesizing something else, and you're synthesizing something besides tryptophan, what if the product does not?
Then you have a problem. If this doesn't work, you're going to have a problem. Okay? So in this case, this is what's called a repressible operon, because we can repress it. It's not currently repressed. Its normal state is being on, but you can shut it off if you need to. Okay? Where the other one, an inducible one, was what? Normally off, but you could turn it on. Okay? So let's compare that here. I don't know whose turn it is. Whose turn is it now? Young? Okay. I've already taught you this, so this should go pretty quick. Okay? Young, you're going to handle the first slide, and Old, you're going to handle the second slide. Go. Okay, go.
The lactose will
It's when the inducer goes through pressure and it gets the repressor.
so that once the RNA is fine, the genes are transcribed.
Is your brain wrapping around it?
Yes.
Okay, yes. I can't hear.
So is this one prokaryote?
I cannot hear because of the rude children. Go.
So is this one where they create like the thing that the repressor binds to? Is this for prokaryotes and the other ones for eukaryotes?
Okay, so for eukaryotes, we're more about positive control, which I haven't taught you yet. Okay, so here you can see RNA polymerase binding to the promoter. Okay, just so your brain can start to grasp it, let me show you something. Let me show you something in our future. Look at this. Okay, wait for it. Okay, if you look here, here's the promoter, right? RNA polymerase would bind to the what? Promoter. You can see there's not an operon here. You don't see operons usually in eukaryote. That's a prokaryotic thing. But this RNA polymerase, he won't bind onto this promoter, okay, unless you have a series of other things that have to happen. This thing is called a TF, or a transcription factor. You have to have an activator that binds to an enhancer, okay? Then the transcription factor binds to the transcription activator that's bound to the enhancer in the house that Jack built. And then once that's there, it folds it back over and helps RNA polymerase bind to the promoter. So if you look at this, you have to have this and you have to have this. And if you look at this, this has 1, 2, 3, 4, 5 components to just that. So if one of these components is missing, then your transcription factor isn't functional. It doesn't even matter if the activator's there. This isn't going to happen. So as eukaryotes, we create a chemical climate in our cells for turning on certain genes. So if this, and this aren't there, those genes won't be turned on. So we're about positive control, helping the RNA polymerase bind. We are not about the operon and negative control. That's more of a prokaryotic thing. Okay, questions on that? All right, so let's go back and let's write notes about both of them. Okay, so we'll start with a repressible one. Okay, and the operon is on. Look on your notes. Operon is number 1. See the trip operon? Did we get the first box? No? Okay. Bacteria's environment is always changing, yeah, okay, just a minute. Oh, so then are the necessary proteins. I can see how I was saying that. So then are the necessary proteins. An operon in bacteria, the genes are often clustered into units which allow the expression of several related genes to be controlled as a what? Unit. Do you see how I italicized that? That's important. Okay? You're controlling them all as a unit. Now, the operon includes the promoter, the operator, and structural genes. I just put in brackets the regulator gene so you would know how that relates to it. The regulator gene is normally outside of the operon, and it codes for a what? Repressor. Codes for a repressor. The promoter is where RNA polymerase initially binds. Where RNA polymerase initially binds. The operator is the region where the active repressor binds. What are you going to put in that word? What are you going to put in that spot? Something RNA polymerase. What does it do? It preventing, good. Preventing RNA polymerase from binding to the neighboring promoter. Blue tells Slade about the structural genes.
Structural genes code for the enzymes and proteins that are involved. Where did you get your key? I can't see what's going
Okay. All right, so we had 2 scenarios here, an operon being on and an operon being off. So the trip op, what do you think that TRP stands for?
Tryptophan.
Yes. The trip operon is classified as a what? Repressible operon. Is it currently being repressed? No. Okay. And so the operon is on. You have an inactive repressor, but it can be what?
Inhibited.
I don't want the word inhibited.
Oh, you don't want
No. What's another? It can be what?
Activated.
Activated. Oh, what can be inhibited is the operon, is what the way I was writing it. What could be inhibited is the operon. Okay, but you can activate the repressor. Okay? Structural genes code for 5 enzymes required to synthesize what? Tryptophan. If tryptophan is present, it acts as a what? What did we call it? Starts with a C. Co-repressor. And it will activate the repressor, turning the operon off. This is typical in anabolic pathways. The product activates the repressor. The product activates the repressor. Okay, so on your number 1, if you want to help on the number 1, where it says the operon is on, the inactive repressor, but it can be inhibited, the it in that is the operon, is the it in statement number 1. If you wanted to do it that way. Okay, now let's do the lac operon. This is an inducible operon. This operon is what? It is off. Okay, number 2, when only lactose is available, then the structural genes that code for the 3 enzymes required for lactose metabolism are produced. Lactose binds to the repressor and inactivates it. Lactose acts as an inducer because its presence is required in order to express a gene. This is typical not of anabolic pathways, but what? Catabolic pathways. Did you guess that already? I'm so excited. Okay, you got your clickers on? Here we go.
Oh my god. It turned off.
What can you do if it turned off?
Turn it back on. I want to be the first one to wait, what is the number? 442. Okay, geez. Why so now? I think we gotta like sleep some more. I get like the most tired. I don't know like ever. You're a lame. I'm a lame.
What's going on? Why are there so much little amongst you
I don't know
guys?
what's wrong with her. It's so rude.
Doesn't even go here.
Wait, what?
'Hey, let's go, guys. Move it. Focus.
Oh no.
Okay, let's talk about it. Where does the repressor bind? To the operator. We had a few of you say to the regulator. What is the regulator gene? Yeah, it's the code book to make the repressor. Okay? And then one of you said structural genes. No. Does anything bind to the structural genes? No. RNA polymerase reads the structural genes, right? Okay. Check your BioBuddies clicker right now. If they don't show, go ahead and ring that bell of shame for them. Or gong if they got it right. Yes.
What's
Allolax. It's the more specific name.
Would that be the
in space? What?
Would that be the inducer?
Yeah. Five, four, three, two, one. Okay, guys, stop and listen to me for a minute. Did I just go over the answer to this question? Okay. Is there any excuse for missing this? Okay, you look at your BioBuddies clicker right now. You apply shame, if need be. Okay? Or, listen to me, ask them, where is the disconnect? Ask them, where is your disconnect? Where are you not getting it?
My disconnect is not paying attention when I do. I keep making mistakes.
That's the worst mouth
Talk to the hand. That's what Sheldon does.
Would they do that?
Oh. My God. There's no other. Guys, who binds to the repressor making it inactive? Inactive. What? Lactose. Okay? So, these are the kinds of questions you have to be able to differentiate between. Because when they ask you questions about this, they're going to give you like several, and on my test, will give you several different scenarios, and I'll say which one is incorrect. So you have to be able, you have to know the vocabulary, you have to understand it, because this is all just negative control. We still have to talk about positive control as well. Read it carefully. This is varsity. Okay, so where are those enzymes? The enzymes are the structural genes when we're talking about the lac operon. Okay, so let's talk about the lac operon. The lac operon is referred to as an inducible operon, right? Who's our inducer? Lactose. Is the operon on in an inducible one? No. RNA polymerase cannot bind to the promoter because there's a repressor on the operator. RNA polymerase cannot bind to the promoter because there is a repressor on the operator. Do I need these structural genes right here? No. What do these genes code for? Lactase. I don't have any, but now all of a sudden lactose is here. Lactose will pull off the repressor. Pull it off.
Oh.
Pull off the repressor because now lactose is here. So now RNA polymerase can bind to the promoter. Boom, boom, boom. Make your mRNA. Right? Make your protein, which will digest, right, this lactose. So if the lactose is all gone, now the repressor can get back on the what? Operator and turn everything off again. Because I don't have any more of that lactose. Now, that is true in a catabolic one because I've got something to digest. If I have something I want to build, and I need this all the time, RNA polymerase is constantly building her, okay? When I have enough of her, she will activate the She will activate, and stay there. She will activate the repressor and turn this off because I have enough of her. But if she goes away, and I make her again, okay? Do you have that?
Yes.
Okay. Okay, now let's level this up just a little bit, okay? And what did I tell you? I said that bacteria would prefer to eat what? Glucose, okay? Bacteria would prefer to eat some glucose. So, now, there is something that bacteria will make when glucose levels are low, okay? What they will make is cyclic AMP. Interesting. Adenine, here's a ribose, and just what? Phosphate. One phosphate. You know about ATP. You know about ADP. Welcome to AMP. What does the M stand for? Mono. And what kind is it? Cyclic. Do you see it? So when glucose levels are low, you tend to have cyclic AMP. Camp, because they ate all the glucose. Now, watch this. This is something similar in eukaryotic cells that I was showing you about just a little bit ago. This is a transcription factor. It's called CAP. Okay? Normally it is inactive. Okay? Normally it is inactive. But if cyclic AMP is around, and why would cyclic AMP be around? Because what levels are low? Glucose. Okay, so if glucose levels are low, cyclic AMP will show up. Cyclic AMP takes your inactive cap and makes it what? Active. When it is active, it helps RNA polymerase bind to the promoter. Okay? Bind to the promoter. Let me ask you this. If there is glucose, will there be any active transcription factor here? Will there be any active cap? No. If there's glucose available, nobody's helping RNA polymerase bind. Do you agree with me on that? But if there is no glucose, are you going to help RNA polymerase bind? Yes. Does it make any difference? You have to have what, though, to even want to make these in the first place? You have to have the presence of what? Lactose. If you don't have any lactose and you don't have any glucose, right, this is still not going to happen. It doesn't matter how helpful that Boy Scout wants to be, okay? There's no lactose. There's no old lady to help cross the street, right? It does not matter, okay? So you have to have the presence of lactose, because if lactose is here, then the repressor will be gone, and then you can even do this. So let's look at those different scenarios, okay? In this case, there is no repressor here, right? No repressor is here. Then you could then bind on here, but it can't bind very strong because what? Yeah, and if this is inactive, that means what is here? Glucose. So we don't want this anyway, because what do we prefer? Glucose. Do you see the chemistry? You only make that lactose digesting enzyme if lactose is present and glucose is not. Because if glucose is present, that's what you're going to want to make enzymes for. And that's how an E. coli can have preference about what it metabolizes. It says, yeah, I'll digest lactose if I have lactose present and there is no glucose. It has a caveat. Okay? Yes?
I had a question. So this is like, I was talking to Tristan, I was like, this is like a real chicken egg situation. Like, which
Yes.
came first?
Which one?
Yeah.
I would assume glucose was more readily available first, and that was synthesized first, and then modifications happen for other sugars. So maybe that's why it's there. Look at these different scenarios. They are not going to freak you out. Okay? Let's look at these different scenarios. If you look at these columns, is there glucose, is there lactose, plus or minus? And then what's going on to our operon right here? Remember, this is your cap binding site. Here's where RNA polymerase wants to bind. Here's the operator. Here are your structural genes. Let me blow each of these situations up. Okay, so let's just talk about scenario number 1. I would like it if you owned it. Instead of me teaching it, you will own this information better if you understand it. So Slate's going to go first. Explain this scenario here on top. Go ahead. Keep in mind, glucose is yellow and lactose is in the purple column.
So glucose is glucose and lactose is present. So if they're both present, then the operon is off. Because cap is the just started the second
Okay, so let me go through that with you. Hopefully they were able to talk. If glucose is present, okay, if you have glucose, then you don't have the helper. You only have the helper there, the active cap, if glucose is what? Yeah, not there, okay? So you have no helper, okay? And lactose is present, but you're not getting RNA polymerase to bind to that promoter because you don't have a helper. Is there a repressor on here? No. Why isn't there a repressor? Because you have lactose. There's no repressor there. Now let's look at scenario number 2. Blue, this would be your turn. Go ahead. Make it big again. Okay, so riddle me this. Do you have a helper? No. You don't have a transcription factor helper. Why? Because you have glucose. No need for the helper. Okay? However, I do have a repressor sitting here. Why do I have a repressor sitting on the operator? Because there's no lactose. So I'm not going to transcribe this because A, I don't have any lactose, and B, I don't have a helper anyway. But it doesn't make any difference. Okay? Because there's no, this right here, this negative control is always going to shut it down. Okay? So now let's look at our next scenario. Whose turn is it? Slate. Go Slate. What's going on here?
Any lactose, so it's still off.
Do I have a helper? Yeah, I have a helper because why? I don't have any glucose. So this little green piece of pie right here is that new one I taught you about, which is what? Camp. Yeah, cyclic AMP. So that's because there is no glucose, because they ate it all at their camp. Okay, so now I have cyclic AMP, which activates the cap. So it's bending the DNA, making it like, come on, RNA polymerase, land, land. And it goes, I can't, because there's a repressor. And it doesn't do any good to land anyway, because there isn't any lactose. So I know you don't have your favorite glucose, but hi, we don't have we don't have a secondary thing either. We don't have lactose, so let's not even make these genes. Yes?
So the cap is useless if there's a repressor.
Exactly.
Okay.
Yes.
The cyclic AMP come from?
The cyclic AMP shows up in a cell whenever you have low glucose. That's
Is it
it.
just like a protein?
No, it's adenine and it's right. Yeah, it's a single phosphate. Yeah, so you just have levels of that build up when you don't have glucose. Okay, now next scenario. Whose turn is it? I wonder what's gonna happen.
Okay, so if you don't
Yeah, because now it can go because it has
Okay. I don't have glucose, so I have a helper, right? Because there's no glucose. So now I want to help lactose because I don't have any glucose. I'm helping. Okay, I'm helping, and RNA polymerase is able to get on the promoter because who is gone? And why is the repressor gone? Because I have lactose. So now I can transcribe and translate and make the enzymes because A, I don't have any glucose, so I need something, and B, I do have something, lactose, so let's digest it. Okay, so that creates that scenario. Okay, so these are both activators and repressors working to maintain homeostasis. Look at your notes. Further control of the lac operon. Number 1, bacteria prefer to metabolize glucose. If there is no glucose available, then cyclic AMP will build up. Number 2, cyclic AMP will bind to who? Cap. Activating it, and it will bind to the DNA, bending it in a way to allow who? RNA polymerase to more easily bind. This process ensures that lactose is only metabolized in the absence of glucose. In the absence of glucose. Types of control. Negative control is anything related to a repressor. Active, inactive, when active, inhibiting transcription. Positive control is anything that facilitates transcription. When active, it promotes transcription. Now, this is a picture I took from the book and annotated. Do you remember when I had you at the very beginning describe where all the areas we could exhibit control, right? So here are those areas. Chromatin structure, DNA unwound, so RNA polymerase combined. You recognize these things, right? Histone proteins, nucleosomes. Is it unwound so we can even access it? Transcriptional control has to do with when you have DNA and you're trying to make, mRNA. Post-transcriptional control. Post means comes after. So I've made the mRNA, now what? So this has to do with cap, tail, cutting out the introns, the speed at which it's able to leave the nucleus. That's called post-transcriptional. Then translational control is anything to do with mRNA building proteins. Post-translation, coming after translation, is anything you do to modify the protein to make them active. Do you see how you could write an essay just on the different areas of control? Do you see how up here at transcriptional control, we've discussed that already, transcriptional control, whether or not RNA polymerase can bind to the promoter, either inhibited by a repressor or assisted by a transcription factor. That's the one we have already discussed. Now we're going to talk about chromatin structure, keeping in mind that we know DNA is wound on what? Histone proteins, okay? DNA is wound on those histone proteins. And if we look here, we're kind of getting a close-up to it. Do you see where it says little histone tail? Okay, now look on your notes, go to your notes, introduction. Most cells have complete, we're in eukaryotes now. Most cells have a complete complement of DNA, all the same genes. Different genes are turned on in different cells. This is called what? Differential what? Gene expression. Okay? Slate, tell them number 3. Okay, so back here, first 3 are in the nucleus, right? The last 2 are out there in the cytoplasm. In general, eukaryotic cells don't have what? Operons. But genes coding for enzymes of a metabolic pathway can still be expressed together, even if scattered among the chromosomes, because they all share what? The same control elements. If you have the same way to flip the switch, if you have the same transcription factor, as long as I make the right components, and those are in the nucleus, I can turn it on in this chromosome, this chromosome, this chromosome, and this chromosome, because they have the same on switch. Does that make sense to you? So I can control them even though they're spaced out. Okay, so the first one is talking about chromatin, DNA and associated protein structure. Remember, DNA is wound onto histone proteins. The more tightly wound, the less accessible it is for transcription. Okay, now we have 2 things you can control. You can control the DNA and you can control the histone proteins. We have 2 areas here we have to deal with, right? Because the DNA is wound on the histone proteins. So this is what I'm referring to in number 2. This is governed by 2 chemical interactions. What's the first one I'm telling you about? DNA what? Methylation. The addition of a what? Methyl group to DNA causes it to be more tightly packed and inaccessible. Yes, we've talked about this already. Remember euchromatin and heterochromatin? This is making heterochromatin. Where did we see an example of heterochromatin, ladies? The X, remember? One of the X's condenses into bar bodies. Yeah. Okay. So DNA methylation, when you methyl up the DNA, it causes it to be more tightly packed and inaccessible, heterochromatin. Or histone acetylation. Acetyl groups are added to the amino acids of histone proteins. These are your histone proteins, thus making the chromatin less tightly packed and encouraging transcription. You will find that in what? Euchromatin. So if you attach a group to the DNA, it actually compacts it more, but if you interrupt the histone, it makes it release. Okay, so take a look at this picture. Okay, epigenetic mechanisms. Epi means what? The level above, right? We've talked about you can have a trait because you have a dominant or a recessive trait, right? But it doesn't mean anything if you can't access that gene. I don't care what gene is in here. It could be a dominant trait. It could be incomplete dominance. It could be a recessive gene. If you can't get to it, it doesn't matter. So anything controlling the accessibility of that gene, that's epigenetic mechanisms. Okay? Look at the things that can happen. Development. What the mother is exposed to when she's pregnant during development, right? Environmental chemicals can affect whether your genes are accessible or not. Drugs, pharmaceuticals. Here's one I like. Aging. Whether or not those are accessible or not. And your diet, the food that you eat, can affect whether or not those genes are transcribed or translated. You can see a methyl group. I have a picture right here. DNA methylation. A methyl group, epigenetic factor, found in some dietary sources, can tag DNA and activate or what? Repress genes, depending on if it's getting on the histone protein or on the DNA itself. Look at some health issues: cancer, autoimmune disease, mental disorders. These are all factors that can be contributed to by our what? Environment. You could have good genes that are shut down because of what you're exposed to in your environment. So go to examples. One we have already learned. Oh, here you already know what this is. Euchromatin is non-staining active DNA. Heterochromatin is dark staining. We already learned about bar bodies. So when one of those X's condenses, okay, we can have a mosaic pattern in the female, because depending on which X condenses will depend on what then is transcribed and translated. So go to heterochromatin bar body. Second X condenses in the nucleus. Mosaic, body of a female. Different cells have different active X chromosomes. So that's what an example of that is. Sweat glands in what? Patches, depending on which one is active. Euchromatin, let's see. Let me show you some more on bar bodies. Here are bar bodies inside a nucleus. Okay, I talked to you about, did I talk to you about calico cats yet? Yeah, okay. And depending on which chromosome, if the coloring is on the X chromosomes, only calico cats can only be female, because they're the only ones that have two X's. If it's a male, they'll either get the black or they'll get the tan. They'll be a solid color. But the females, it depends which X is active, which is getting transcribed and translated, which colors you see. So calico cats are always the females. Because people have asked me, they go, wait, how do you make more calico cats then if they're always female? It's the coloring, okay? The females can have the calico, the patterns, depending on which X is active. If it's a male, they either have this one or this one. That's it. Okay? Now these are called, oops, let me go here. So this is showing you it could be tightly packed or it can be unwound. These are called lamp brush chromosomes. Lamp brush is an old school name for when there were lamps that actually got cleaned on streets. You would be more related if I called it the test tube brush, right? Where parts of the brush is sticking out and then part of it is tightly coiled like this. Okay? So here the DNA is all tightly coiled and here the DNA is exposed. That's evidence of activity. Which genes are activated and which ones are tightly closed. Okay? And again, this is all relating, I'm just taking you back to epigenetic inheritance. So on your notes, go down to epigenetic inheritance, inheritance patterns that do not depend on the genes themselves. That do not depend on the genes themselves. And then genomic imprinting is when DNA is methylated during gamete formation. When DNA is methylated during gamete formation. Okay, if that DNA is methylated, okay, if I'm affecting the histone proteins, if I'm affecting the DNA, I am controlling everything that child inherits, even though the genes are there. Depending on if I methyl it or not will depend on whether they can ever transcribe or translate it. So that is something that can be inherited. Okay? All right, transcriptional control, we have already talked about this. Oldest bio buddy, explain this picture. You should have no problem. We've already talked about transcriptional control.
lactose that binds to the repressor or the DNA binds to the repressor and moves it as well.
Okay, and this, your notes, is the most critical level of control. Most critical level of control. Now, in eukaryotes, we leveled up. We said it's not about an operon, it's not about negativity, it's about positive things. So, in this case, we need a transcription factor to help regulate whether that gene is expressed or not. And then this takes us to this slide that I already talked to you about. Okay? So, you have transcription activators that we're seeing here. And the transcription activators are proteins that do what? Speed up transcription. They bind to a DNA region called an enhancer. So the activators bind to the enhancers. Once they're bound, they can help the transcription factors help RNA polymerase bind to the promoter. So it brings a whole loop together. Now, this is simplified. It could be like this. Okay? So you could have this enhancer region, this enhancer region, this enhancer region, and this enhancer region, and all these things have to be connected to this, which helps this get this gene translated. These are like checks and balances. We're not going to make this product unless all of these things are in place. Okay? So on your notes, go to transcription factors. I gave you everything for that. Go to transcription activators. I gave you everything for that. And then repressors. Okay, now, this is now leveling up again. So I'm telling you, I've taught you a very simplistic method of control, but if you look in this situation, these are all different areas where it can be controlled in order to promote that transcription. Anything that inhibits any one of these things will stop that gene from ever being transcribed and translated. Let's go chemistry, cat. Yeah, transcription factors, in my analogy, I would say they're like Boy Scouts, being helpful. Okay, they're being helpful. All right, now go to. Did I jump it? No, I didn't. Okay, go to post-transcriptional control. Okay. Just a second, I'm trying to find where did I jump around here? No. I didn't? Where did I put in your notes about transposons and jumping genes? I know it's somewhere in here, but I don't know where I put it, but we'll talk about it. Okay. So come back to me. What? Oh, okay. Yeah, I don't know why that's sitting right there in that spot, but well, it's okay. We're going to talk about it. It's fine. And then we talk about it in the next chapter too. Transposons are jumping genes. These are genes that literally can remove themselves and re-put them in somewhere else. And this is how you see Indian corn, which we could do a chi-square test on the coloring. Okay? And so when you look at post-transcriptional modifications that take place, so we know about, is the DNA available? Is it methylated, not methylated? Can we do transcription? Do we have helpers, activators? And then we can control which genes are expressed by which introns we move. This would be post-transcriptional modifications. We know also the ability of it to leave the cell, to leave the nucleus, and then this looks like a big piece of turkey here, but what are these things right here? Those are the ribosomes, okay? So this is post-transcriptional control. Being able to do translation would be translational control, and the way this blue protein right here is modified would be what? Post-translational control, okay? Take a look at this picture right here. This has to do with just the way you process your mRNA. So in this case, if we look up here, we have A, B, C, D, E genes, okay? But depending on how we do our splicing, you could still have the whole sequence, or in this case, the second scenario, what's missing? C. So as a result, we have the same DNA, but we have two different products from the same DNA. Now think about this with your large mammalian brain, okay? So we have all this DNA and we say, oh, if I know the DNA, then I know what you're going to inherit. Is that true? No. Because I could know the DNA, but is it methylated or not? Is there something that can help unwind it? And even if I do unwind it and make mRNA, which of those genes are going to be remaining and which ones are going to be cut out? That's where a lot of the control is. And that's where a lot of the studying is taking place. Because, yeah, we know the code book, but this one section of DNA can code for so many different things. Yes?
Is that why identical twins sometimes don't look alike?
Exactly.
No.
Now, go to your post-transcriptional control, differential excision, removal or inclusion of introns and splicing, and example DNA coding for hormones. You can have DNA that makes a hormone, it could make an enzyme, that same DNA. You could make an enzyme that digests proteins, or it could be an enzyme that makes a hormone. Two totally different things coming from the same DNA. The speed of transport of mRNA through the nuclear pores. And then sRNA. S stands for small. And these are transcribed RNA loops that become what? Double-stranded RNA. Now, is RNA normally double-stranded? No. Okay. And so, this double-stranded RNA is cut up to form small RNAs. These small RNAs can regulate both transcription by binding to the DNA and translation. They are the source of microRNAs, miRNAs, that can bind to mRNA, inhibiting translation. And this is right here where I have the microRNAs. Okay? And small interfering RNAs form a complex with an enzyme, RISC, and degrades any mRNA the complex is complementary to. What, Blue, tell Slate what RISC stands for. Why would you say the RNA was silenced? Because you destroyed it. Yeah, you destroyed it. Boom, you're done. Okay? So, these are kind of, I'm talking about the RNA, but it actually is influential in several of the different steps, because if you do your DNA unwinds and unzips, and you're ready for RNA polymerase to bind to the promoter, but some other little fragment of RNA binds on there, do you see how that could interfere with it? Then it could interfere with, I've made my mRNA, I'm cutting, I'm splicing, I'm trying to make it leave the nucleus, and it wants to go get translated, but it can't because another piece stuck on right there. Okay? Or it can activate other RNAs that end up cutting this mRNA up. So it can be used in several different spots. Okay? Go to translational control. I gave you everything for translational control. Old Bio Buddy, you're in charge of that box. Go. Okay? And then post-translational control, following protein synthesis, additional changes. How long does the protein last? Are there some sort of enzymes that are going to digest it? So go to post-translational control. It is only considered a functional protein if it is what? An active gene product. It doesn't matter if you've allowed everything else to transpire. You have transcription, you have translation. It doesn't matter unless you can take that protein, fold it in the right shape so it can be active. Okay? So proteins are continually being formed and degraded. Only keep what is needed. Okay, and then I gave you the rest of that. Okay, I got to talk about this last part, okay, and show you some scary pictures. All right. We already know this part. We have actually already talked about this, okay? You know that here's your DNA. Here is the codon, right? Here's the messenger RNA. A with U, T with, and G with C. If you have a tRNA land on this particular mRNA, okay, UAC, you know what the tRNA would be. The amino acid it brings is what? Tyrosine. Happy, normal. If, however, the DNA gets mutated, instead of ATG, you are ATA, this now changes our codon. Now, instead of UAC, it is what? UAU. But guess what? There's more than one way to skin this cat. There's more than one way to make tyrosine. It just so happens that both UAC and UAU code for tyrosine. So this is referred to as a what kind of mutation? Silent. We're not stressed about it. Here, though, instead of ATG, it's been changed to ATC. We have a different codon, and this particular codon is a... So you're right in the... I was in the middle of my sentence and I stopped. Okay? So now you make a protein, okay, that is what's called a nonsense protein because you're building your protein and all of a sudden you stopped in the middle of it. Okay, and in this case, you have a mutation, a different codon, and it codes for the wrong amino acid, and it's a missense. It makes no cat in the middle of your sentence. So I put something in there that was wrong, right? And now it doesn't fold correctly. You have a missense. And that's an example of that is sickle cell disease. In sickle cell disease, because you have the wrong amino acid, it folds wrong, so you don't have this biconcave disc for a red blood cell. You have this different shape, and it's sickle. Okay? This is the worst one of all. This is a frame shift mutation. Now, codons, I know you don't have the codon chart memorized, nor do I, but I wanted to pick 3-letter words that you would understand. So if you look at this, you could read this, right? So I'm going to space it out a little bit. The big fat cat ate the big rat. Now, if for some reason I get rid of the E due to a deletion, now my first codon is not THE. What's my first codon going to be? TH what? B. And it throws them all off. So now it says thiga ifa attic atatet heba igor at. Okay? So by removing one nucleotide,
Today, nutrition, the University of Delaware will be in the College and Career Center.
I threw them all off. Okay? So look at your notes. I want you to know. Let's fill in that last part there, because okay? Go to introduction. Define permanent change in the sequence of bases. Permanent change in the sequence of bases. The impact from no effect to complete inactivity. On 3A, your word is future. Transposones, causes of mutation can be spontaneous transposones. I've already talked to you about. 1A, put the word jumping in there. Number 2, induced, may result from exposure to toxic chemicals. We have already talked about mutagens, so you can put changed DNA. on point mutations, it involves a change in a single DNA nucleotide. Single DNA nucleotide. And this last one I just taught you was frame shift. As a result of an addition or deletion. And then all the rest of the notes you have, which you can read through. And you have this presentation. This was the fun picture I wanted to show you. What? Okay, and when am I going to review this chapter? When am I going to review it? Monday. Monday. You have all your notes though, so read through them and make sure to look at the presentation if you want, and if you're looking at this late at night, eat toast.